entomology, which need to be studied in JRF also and other exams also. Some message is coming on my screen. Please move this window away from the shared application.
Some students, they are trying to modify your screen or whatever it may be. Just don't modify your setting, because now
Okay,
your
sir.
personal.
We will continue.
Okay.
Some disturbance is there. I don't know. Maybe they are not doing, but maybe some setting. Okay, let me share once again slides. So you know that this whole exam is now computer based test, and the duration is for JRF. Duration is 2 hours, and total number of questions are 120. All are multiple choice questions. Like in SRF also you have multiple choice questions, but for ARS you have 1 marker question, 2 marks question, like that 5 marks question. You have there you have a different pattern, but syllabus is also almost same. So scoring pattern for JRF, if you see, there are 4 marks if you are giving correct answer, minus 1 mark if you are giving incorrect answer, if you are attempted and it is an incorrect answer, and if you are not attempting at all, then no issue, that is 0. Then paper is in English medium, and what I wanted to give here is approximately, if you see the average of if you see some last 5-6 years question papers, then in 120 questions approximately you get 65 to 80 questions from entomology, 20 to 35 questions from nematology, and 15 to 20 questions from general agriculture. So because I was asked to speak about entomology, now we will start discussing about entomology. So first part is as per the syllabus, if you see, so you have to know the introductory part of entomology. So introductory part includes its importance, its classification of phylum Arthropoda. Now classification, if you see from phylum, it is not just orders and species only. You have to start from Arthropoda, classification of Arthropoda. Then you should know when the what you can say this year wise, means in what happened in the 600 BC, which that phylum came, these subphylums are there, which insects came in which era. That more information is there in Entomology at a Glance book. If you see Entomology at a Glance, in that book they have given nicely how this history or the classification of this phylum Arthropoda with its origin. So where the insects originated, that period, and all these details are there in Entomology at a Glance. That book, if you see, you will find that. Then reasons for insect dominance. These aspects might have been covered in your syllabus also. So not much is there to say about this. Then history of entomology in India. So in history of entomology in India, you must know the starting of entomology when it was started at Pusa Institute in Bihar. So who was the pioneer? Then Pusa Institute was shifted to the this Delhi. Then about Fletcher, what is the this your contribution of yes Pradhan. So Pradhan sir is considered as a very big personality in our entomology. So you should read about the Dr. Pradhan also. I think it is there. Some information is there in our website also. If you see IRI website in entomology section, if it is not there, whatever I remember, we have uploaded there. If it is not there, you can anytime contact me. Now your sir is having my contact number, so we can provide this information also. So not only history of entomology, but now you slowly you have to start with the history, and you have to now you should know which are the major institutes in India who are working in on entomology. Like NBAIR, which is located in Bangalore. Now with that name, we should know what is the current name. Most people they call now NBAR. Now it is name is changed. What was the earlier name? That also you should be aware about. Like institutes like NCIPM, it is totally for plant protection. About that institute, what is the full form of NCIPM, where it is located, and then such other institutes which are mainly in entomology that you should be aware about. And in general agriculture, they can ask any institute. So you should know these central institutes, the different these research centers and other institutes of ICR. So some points, if I want to discuss with you regarding the your history of entomology. So in ancient script, the Ramayana and Mahabharata, some books you can get where they have given some information in whatever found in Ramayana and Mahabharata also. But I think for this section, you follow this in entomology at a glance book. There, very important and related information is given. So what I have tried here is, after each section, I have tried to give some questions which are memory based, which I have as a memory based questions, which we were been asked earlier in JRF and SRF exams. So what type of questions are there, then you will be knowing from this. So you can see here, who is considered as father of taxonomy. So when we study entomology, you know, taxonomy is another one section, then toxicology section is there, IPM is a section. So who is considered father of taxonomy? Then which edition of Systema-Naturae contains binomial nomenclature? So that you should be aware about. Who wrote this Systema-Naturae, you should know. Then DIPA, what is this DIPA? Then DPPQS, this is another institute you should be aware about. Under the DPPQS, we have Central Insecticide Board and Registration Committee, CIBRC. So what is the function? I expect that students are noting some points when I am talking. So in case of DPPQS, you know, CIBRC is under DPPQS, and CIBRC looks after registration of insecticides in India. So there you will find more information that I will be discussing in next some slides also. Then about Central Insecticide Act, because this is all part of our history in Indian history also, that what is the rules and guidelines in India. Now new management bill is new, this pesticide management bill 2020 is now going to come. So you should know about this act and its rules, when these rules of this act came into force, and this 1968 year for this act. So next section is systematics. In systematics, mainly you have to focus on definition part. There is one book, Animal Taxonomy by Kapoor. So in that book, very nicely that definition part is has been given. So that book you can refer. It is there everywhere in libraries. Then you should know what is the ICAR-AIEEA. I think you will be aware about that, International Code for Zoological Nomenclature. Simply they can ask this full form for this word, and they can give very confusing words. So you should be clear what is each word is there. Then classification given in IMS book, that you have to follow. How many orders are there? But I think one order has been added now. So that classification, you know, and here when you are studying about the classification of insects, that time you parallel with IMS, because IMS it is not possible for UP students to study IMS book. So what you can do with IMS, you can keep Insecta book. You might have seen the book Insecta, that is published from Tamil Nadu Agricultural University. So you can keep that book along with this IMS book, then it will be easy for you. In Insecta book, they have given very clear and very easy to understand and very important points they have covered. That in each order, for each order, what is the synonyms? What is the meaning of that order name? Like if it is a ortho, this your what is called any Odonata, what is the meaning of Odonata, why that name is given, and is there any synonym for that order, that also you should be aware about. Second thing, once you know synonym and it's why that name is given, then you should know some examples which are confusing actually. So by name it will look like that insect is in another order. So that examples also they have given nicely in Insecta book, that you have to remember. Then other parts, when you are studying initially in UG itself, you are studying all morphological parts. But when it comes to peculiarity, some insect have peculiar wing venation or leg modification, modification in thorax or modification in abdomen. So that peculiar modifications you should know along with our general morphology of the insect body. So some previous question I have given, like high in this for this section, that high, which is the highly evolved order, which is the order which is connecting link between like Lepidoptera and other next suborder. So that everything is there in IMS book and your Insecta book. So you should read that book very carefully. So other questions I think you can go through. This presentation I will be giving to sir, so they can share with you. You can go through how what type of questions are being asked. Like here you can see asymmetrical cerci are found in. So like these characters are typical for any order, for that order or in even families. Some families of parasitoid insects are typically known for their modification in wing venation or modification in their leg segment. So that type of typical things you should remember. The morphology I have already discussed with you. So here what I have included here is that is general part what you are studying in UG also. In morphology, I think that course. So basically when you are starting to study all those what I have previously discussed here, before that you have to go through this morphology course, so that you will be first you will be aware about all the naming and all the parts of the insect body. Then you can go to the modifications. So what are the major segment to which we call tergum, sternum, pleurum, that all you should be aware about. Then the definitions like these are the words like molting, ecdysis, stadium, instar. So usually they will confuse you in the exam about these words. So you have to understand these terminologies. What is this terminologies for which it is used? So this you can find in your books and your notes also. You can go through. So for this particular morphological type, here you can see questions are there on mouth parts also. So in case of typically insects like your Thysanopter like thrips, what is the modification in mouth part? In mosquito, what is the modification in mouth part? So all these mouth parts and their example, within that if there is a modification, suppose there is a chewing type of mouthparts and there is any modification there, then that also you have to remember. So like wing modifications like halters, fringed wings, elytra, autogmina, these all modifications you have to understand and you have to remember. Then insect anatomy and physiology is another section, very important section. So in this section, usually you know about the system, about the systems like digestive system, circulatory system, respiratory system, excretory system. Now, when you are studying for your competitive exam, what you can do, along with your notes, you can take Pant and Gai book for this particular, for this anatomy and physiology sections, the systems, insect systems, various systems. So along with your notes, you take your this Pant and Gai book. I think it is available in every library now. So in that book, very nicely they have covered the modifications in each system. Why that modification is there? Not only modification, why that modification is there, that is also required. Hello? थोड़ा क्लास में हो. अच्छा ठीक है. So what happens? Suppose one cryptoneopredial condition is there, and when you are studying this condition, you have to understand why this modification is there. What is the functional significance of this modification? So then, even if in exam, if you remember this cryptonephridial by just mugging the name of any insect, then in exam, what they will do, they will give another some type of another insect which is of that same type. Then you will get confused whether they in that insect that type of modification is there or not. So first you have to understand what is the modification, why that modification is there, means it is functional significance. And once you know that, then you have to see the examples. What are the possible examples given in your notes and your books? So I think for this all these systems, I always followed Pant and Ghai book. Some aspects you can see in Chapman books also. Chapman book. So if Chapman book you can in UG, sometimes I think it is tough. If you have time, you can go through, or you have to take help of your seniors, so that because Chapman book is big, you cannot go through each and every chapter. So important in Chapman book is these systems. So Pant and Ghai book and Chapman book will be sufficient to cover all the modifications and basic aspects of these systems. So some examples I have given, like filter chamber. It is a modification of alimentary canal in Homoptera, in which anterior portion of midgut is closely associated with the hindgut. Now this is the general. First you should know what is the filter chamber. Now next you should know what is the function of this filter chamber. Why this modification is there. Now once you understand that, then it is, you know that it is now in Homoptera. So if even if somebody gives the name of the insect, that filter chamber can be found in following type of insects, and if they give the name of the insect, then you should know whether the all insects are there in Homoptera or not. If there is an exception in that, that also will be there in book, that you have to understand. So likely cryptonephridial condition, like I was telling you, diastole, systole, then prophylaxis. What is the prophylaxis mechanism? Where it is found, which insects they use this prophylaxis, what is the purpose of this prophylaxis? You will find it in even Pant and Ghai book, even it is in Chapman also it is there, even in IMS also it is there. Then dorsal blood vessel. What is the dorsal blood vessel? What it is function? Because by name it looks something different, but now you will be aware about, because you are studying basic courses of entomology. Then what are the accessory pulsatile organs, and is there any modification? Is there any functional differences in the different insects? So likewise, some other information I have included here, like when you are studying the nervous system. So what is this axonic conduction? What is this synaptic conduction? Which chemicals are important when synaptic conduction is taking place? What is the chemical or what is the enzyme which is responsible for breaking of this choline acetylase or acetylcholine? What is the function of this acetylcholine? So all these aspects you have to understand when you are studying particular system. Then which gland secretes or excretes which type of hormones? That also you should know. Now it is about hormone, then you should know about the pheromones. What type of pheromones are there? I think that I have included in some slides, that also we will see. Then type of ovarioles, when you are studying the reproductive system, there are also some variations in ovarioles. That this is just an example I have taken. So what type of ovarioles are there, where we find this type of modifications, that you have to know. All these details are there in Pant and Ghai and Chapman. So types of reproduction, like oviparity. What is oviparity? What is viviparity? What is ovoviviparity? And now once you understand these terms, then you should know the examples of it. Where you find oviparity, where you find viviparity. Now in aphid, you take an example of aphid. Everybody of you should be clear about the aphid life cycle. Like we study this honeybees, their life cycle, their caste system, in ants, in termites. We study everything. Like that in aphid also we have alternation of generations. So how it is happening? If you see the KP Srivastava book, if you have, that is title, I think I just forgot that. KP Srivastava is the author. So in that book, very nicely that life cycle of aphid is given. So there you will understand this all these terms. So and this hemophilic viviparity, pseudoplacental viviparity, adenovirus viviparity, all these types of viviparity and their modifications have been given in Pant and Ghai book also. Then this other like respiratory system, if you go to respiratory system, there are certain modifications. In some insects you will find blood gills, then caudal gills, rectal gills, tracheal gills. So first you have to see the respiratory system, then you have to find out where these modifications are happening. So all these modifications and their functional, why these modifications are there, that you have to understand. So another things I have given you, insect defense. In case of insect defense, there are some terms that you have to understand, that Thanatos a hard four-wing, then mimic or nature, then other things like mimic and mimicry. Mimicry is also one aspect that also you have to cover. So on this section, some questions I have given. Suppose when you are studying a digestive system, they will not ask simply that what is the middle part of digestive system or anything. What is called as, what is another name of midgut? They will not ask like that. They will ask like these questions, like midgut is dot dot origin. Whether it is a ectoderm, endoderm, mesoderm, or none will not be given. They will give something confusing here. So you should know why we give these terms. First, you should understand this. It is there in your now courses, UG courses also. So first, you have to understand why we called it as endoderm, ectoderm, endoderm. That you will understand in your embryology part. When you are studying embryology, and in IMS book also, they have given whenever these systems are there, there they have mentioned this. Each part, what is their origin, and why it is like that. Why some parts are ectoderm, we call ectoderm origin. Why some parts we call endoderm origin. So you have to understand first these terms, and then you have to see which system and which part of the system has which origin. Then now see peritropic membrane is present in. Now this is everybody of you know that peritropic membrane present in midgut. Why it is there, that maybe you are knowing. Now even though it is important in some insect which you know, whether it is absent in some insect, whether it is absent, that exception thing, exception examples also you should know. So they will ask questions like peritropic membrane is present in aphid, whether it is true or false, like that. Means some question they will form and it is there in aphid or not. So you should know what is the peritropic membrane, why it is there, and then whether it will be there in aphid or not. So simply you can guess whether it will be there or not. Then filter chamber is present in, like I told you in earlier slides also. Like that, you have to prepare yourself. Then ecology part. In ecology, some actually in UG for JRF, not much depth. They will not go much depth. Whatever you are learning in your classes, like initial definitions of ecology, there your trophic system, then food wave and food chain, then social life, like honeybee, social insects or social life related phenomena. All these things are enough for the ecology part portion. Then again here important terms are like dormancy, hibernation, estivation. What are the difference between these systems? Which of these are which of these are innate characters, means transferred through the gene, or which are through just driven by the climatic situations. So that type of differences you should know when you are studying this dormancy, hibernation, estivation, that type of phenomenon in insect life. So is there those phenomena are happening particularly extreme in case of some insect, that also you should aware of. So in case of sampling, I have tried to give some introductory part. You can go through and maybe more is covered in your courses. So it is not possible for me to cover each and every aspect here. So what is dormancy? What are its type? Quiescence, diapause, even hibernation, one another word is there. So what is the difference? What are the synonyms? That also you should know. So some questions I have given like earlier sections. Then biological control. Biological control usually when we are studying in UG classes, it looks very easy. But when you go for competitive exams, there they will ask very peculiar terms. So in biological control, you have to be sure that you are through with all the terms which are used in your courses. Particularly when we were UG students, there was one special course in last semester. I don't know current situation. So in that course, usually that part was covered, like what type of parasitism and all these details. So that course, I think they are now merged with earlier courses. So here, history of biocontrol from DBAC you may be DBAC scientist, you may be knowing now. So what is the history of biocontrol? It is given nicely in KP Srivastava book. That will be, I think, sufficient. Or even your entomology at a glance, जो सक्सेना का book है. In that also they have given very good explanation or the questions on this type of this biocontrol. Then organizational setup of biocontrol in India. Right from PDBC. You should know the PDBC and now how it is a NBAII. So from PDBC, what name was changed, what is the current name, and any other aspects of biocontrol in India. Then introduction to biocontrol, that course you are aware about, that course you should be through. In that you know that terms like inoculatory lead, inundatory leads, then introduction. What are the example, very peculiar examples of introducing biocontrol agents from other country to our country, right from history till date. So we should know for till date for even I think for papaya, mealybugs, and after that some insect have been introduced, biocontrol agents have been introduced. All those details you should be aware about. Then insect diseases also very important. I think there is special course on this or some part is covered. This part is there in KP Srivastava book also. Even in insect at a glance book also they have given nicely what are the different insect diseases, what are the causal organisms, and what are the different symptoms or what are the synonyms, green muscardine terms that you will get in this the course or in these books. So some terms I have tried to enlist here, so you can understand what type of terms you have to study, like predator parasitoid, you know. Even some students they get confused in parasitoid. Many times they have used this parasitoid word to ask questions many times. So for the definition, you see the definition of this parasitoid word in Triple E book. That Triple E book you may be knowing that elements of economic entomology. They have given very nicely this parasitoid and this terminologies also. They are also very nicely discussed. Even I told you in insect at a glance, that book also. So solitary parasitism, super parasitism, kleptoparasitism. Most of the time this question is asked. What is this kleptoparasitism? What is this adelpoparasitism? So these terms you should be aware about. So these principles of biological control you might be studying in your classes. That is classical biological control, augmentation, and conservation. So these examples, first you understand the concept of these three and important examples which have been takes place in India and abroad, means which are the important phenomena which have happened in the entomological history, you can say. So that you should be aware about. So some questions I have given here, you can go through. So next part is chemical control. Chemical control is here your insecticide, mainly insecticides and the new generation pesticides like growth hormones and all. So here you have to study the History and Insecticide Act Insecticide Act, it rules and who is framing, who is responsible for this Insecticide Act, or who is responsible for registration of insecticides in India. Like I told you, CIBRC, Central Insecticide Board and Registration Committee. You should know about that also. So insecticide formulations, I think I have tried to include some slides also. I will be giving some PDF files also. So that also you have to go through. First, you have to understand in case of insecticide formulations, why we formulate insecticide. First question. Then, what are the components of insecticide formulation? Active ingredient definitely will be there, and then other inert ingredient. What are the other inert ingredient are there? Then the short forms used for these formulations, like you might have seen EC. EC means emulsifiable concentrate. EC, SC, OD. So that one file is there. I will be sending that. So you have to know all those terms also. Then classification of insecticides. So classification of insecticides I will be showing in next slides. First is conventional classification, and another is the classification given by IRAC. So you should know what is first IRAC. If you Google it, definitely you will get it. So what this IRAC, they have given a new type of classification board based on mode of action of insecticide. And whatever we were using, another classification like based on the chemical nature, based on mode of entry, that I will be showing in next slide. Those are conventional type of classifications. Please mute whoever. Then another thing which I have missed in the next slide: synergism, antagonism, and potentiation. You must know the difference between these terms. Shortly, I will tell you. Synergism, here your one is insecticide, another molecule is itself of itself is not toxic molecule. When you mix the non-toxic molecule with the your insecticide molecule, it is increasing the toxicity of your original molecule. So you can see A plus B. Here A is toxic, B is non-toxic. Now, if you mix them, A plus B, if you do and you treat insect, then you will get a toxicity or the mortality of the insect, which is greater than the toxicity of only A. So, when you are mixing toxicant with a non-toxicant molecule, you are getting more toxicity. So, it is called as synergism. Antagonism, here, when two molecules are mixed together, the toxicity is decreased. The mortality caused in insect is decreased. And potentiation, here, both the molecules are toxic molecules. Two toxic molecules, when they are mixed together, you will get a mortality of the insect or toxicity to the insect, which is greater than sum of A and B. So you may be able to understand. So you just find out the synergism, antagonism, potentiation words. The clear definitions are there, and you have to understand it. So, and another thing is you have to understand. You have to remember some important synergisms like PBO, piperonyl butoxide, for which group of insecticides it is used. So that also you have to remember. If you're just noting, that is PBO, piperonyl butoxide. So you read about synergism, antagonism, potentiation, and some examples of synergists. They are called as, those non-toxic molecules are called as synergists. Then insecticide poisoning and its treatment, that antidotes. So here you should know the universal antidote, and then antidote for particular insecticide like organophosphates, organochlorines, carbamates, and other insecticides if they are there. That particular antidotes you should be knowing. Then insecticide resistance, mechanisms of insecticide resistance, and how we manage it. Then metabolism of insecticide is mostly, this portion is for SRF. Not much it is asked in JRF. Most Sorry for the pause. So then this metabolism of insecticides, insecticide residues, pesticide hazards and pollution. These mostly this portion will be more in depth for the SRF exam and ARS exam. Not much is there for the JRF exam. Then definitely safe handling of pesticides and insecticide application technology that you may be studying in your courses. Now here when you are studying this insecticide application technology, because nowadays two things are more in focus. Like one is your locust control, because there was a population increase, or you can say outbreak in last year. So about locust you should know everything about what is the significance of neem with the locust, that you have to know. Who discovered the neem antipedantic properties against locust, that also you note down and you have to find out. Then about locust life cycle, and then what are the insecticides recommended by CIBRC for locust control. That is there, that file I will show you. So I will show you where to find that file. That recommendations you should be aware about. Now next thing comes aerial application. In aerial application, what is the peculiarities? Whether we need specific type of this nozzles or formulations, that you should be knowing. That in KPC Vasta book it is given. Next thing is nowadays it is coming up is that drone technology. So in case of drone technology, not much information is there in the public domain. Even truly speaking, we have not been able to understand this standardize the any methods for drone, but whatever information is available, that you have to collect and you have to read for the drone technology also. So that type of, because these are the recent developments that you should know. So likewise, I was telling in the insecticide formulation. In formulation, there are main two ingredients. One is active ingredient, which is a molecule which is killing the insect, and inert ingredients. Other ingredients are added to it to make it a formulation. Now, why we are making it formulation, that you have to understand from your classes. So this is just, I have given some slides where I have noted some historical background of chemical use in entomology, or you can see the history of chemical use in for insect control. So all these slides are self-explanatory. What is the pre-DDT era and what is the DDT era? Paris green for which insect was used? That all the details I have given here. So what is the significance of DDT? Its long form also you should know. What is DDT? What is DDE? It's another analog, DDE. Then what is BHC? What is 99% gamma isomer of BHC? If you are noting down, you just note down 99% gamma isomer of BHC, what it is called, that you should be aware about. So definitely, like those insecticides which are very important in history point of view, that for that insecticides you should know much more, like DDT, its relation to Muller, then what Muller got, where Muller was working, he was working in Gigi company. Then about BHC. BHC, it's a synonym, it's long form. Then, like I told, 99% gamma isomer of BHC. Then, who was responsible for BHC? The identification of BHCs and toxic properties. Then, like organophosphate insecticides, I have given it this era of pesticides in that. So organophosphate started to appear in 1945. So what are the important organophosphate insecticides? So that definitely you will be studying. So when you are studying, you will come across with the insecticides like dichlorophosphate. आप है कि जा रहे हैं अभी बाद में कर नंबर आप कर दीजिए. So when you are studying the important examples like dichlorophosphate, you should know about the status of these insecticides. Now, what I will do is I will be giving you the currently used insecticides. I have given in that list some other details like to which group these insecticides belong, and another file I will give where the status of insecticide is given. So status means, now suppose take dichlorowas as an example. What is the status of dichlorowas? Whether we are using it, if we are using it, against which insect we are using it. If not, if you are not using it, whether it is banned for manufacture, export and import and use, only for banned only for use and manufacture. So all those details you will get in that list. So there are some peculiar insecticides like endosulfan, dichlorowas, borate, other insecticides also there. So for them there is a restriction, like aluminum phosphide or phosphine we use for as a fumigant for storage insect control. So what is the status of that? Whether it is in public use or it is in a restricted use. If it is a restricted use, what type of restriction is there? That restriction you should know. So like these details you should know, and I will be giving that those information. So you go through that files. I think that will be sufficient for you. So some new insecticides like formamidins, hormone mimics, their examples, you should know what are the hormone mimics, then chitin synthesis inhibitors. So when you are going to study this type of group of insecticide, you should be clear about what is the role of these hormones in the insect body. What is the role of juvenile hormone in different stages? What is the role of chitin, this other hormones, so that you will be understand how these insecticides are going to work in the insect body. So it will be easy for you to understand this type of insecticides. So this in short and what you can say in short and with examples, it is given in the one Reddy book is there, short small book is there. Usually we follow that book at the last time, last days of preparation. Means when once you are you are through with your concepts and all, then you can go through like that. DS Reddy top 5 book is there. I have listed that book. So that type of book also you can read for in the your last that when you are approaching to your exams. Before that, in 15 days, that type of book you can study. So because I may miss, that's why I'm telling here, you have to know the status of insecticides or insecticides which are used in storage pest control also. So there are different set of like deltamethrin is used, malathion is used in insect storage pest control as a contact insecticides. Phosphine is used as a fumigant. Methyl bromide is banned for use, but it has restricted use. It is used in ports only. So that all the things you should be aware about. So the classification which I was telling about, that two conventional classification and modern classification given by IRAC. So in conventional classification, we classify them based on mode of entry. So whether they are entering through contact, stomach or fumigant, based on mode of action, based on chemical nature. So these all the things you have covered in your classes. So I'm not going in detail. So next is IRAC. In case of IRAC, what is thoda sa significance kya hai, I have given here. So if you go to the website of IRAC and if you find their mode of reaction classification, so they have given classifications like that: main group and subgroups. So carbamate type of insecticides are acetylcholine esterase inhibitors. Organophosphate insecticides are also acetylcholine esterase inhibitors. So this you should know. So what in exam, what can they do? What they can do? They can ask like simple question is carbamates are which type of inhibitors, that I can ask, or they can give example of any carbamate insecticide and they will ask what is the mode of action of this insecticide. So first you should know that this insecticide belongs to this group and then mode of action is this. So they have given different mode of action. So what you can do is from this list they have given, you can just list out in your notebook. First, you remember main classes for different insecticide groups, and then you remember the different insecticides. So many times when we go through the classes, your EG and PG, definitely you are remembering the which insecticide belong to which group. So you don't have to merge all the things. So you just have to remember that which type of mode of actions are there. Like nicotine, then other neonicotinoids, they belongs to same group, so that you should know. Spinosins, so spinosins are having just some difference of mode of action. So you can see it is a competitive modulator, it is a allosteric modulator. In short, I will do, because it is not possible to explain you. Competitive modulator means the molecule which you are using, it is competing with the natural uh, the neurotransmitters or the molecules to bind to particular receptor. They are competing because they are attaching or at the same site in the receptor. Whereas other molecules like this allosteric modulator, they will bind to that receptor but at the different site. So they are not competing with the natural molecules which are there in the insect body. So that's why they are called allosteric modulators. So like this, you have to know, and for BT type of insecticides also it is given in this list. That also you have to know what is the mode of action of BT also. So some inorganic insecticides I have listed here. So you can see if the some important information is there. So some history of carbamates I have given here. So it was identified first. It was started. The story was started from a plant from Scotland. This Egbo society. There was one Egbo society in Africa. From that society, this history of carbamate started. So they were using that plant for poisoning a person. So from that clue, the Scotland people, they take the clue and they took that seed to the Scotland and they grown the plants. Then they extracted the material from this plant and then they identified that plant as Physostigmine venenosum. Then they identified what was the toxic principle in that extract of this plant. Usually that was a milk of this plant. So they extracted, they identified the toxic molecule. Then the alternate terms came into existence. And based on this, then there was a uh, uh, identification of the you can say the history of this carbamate insecticide came to existence. It's the same. So in case of carbamate insecticides, this carbamic acid, this is a monoamide of carbon dioxide. This is a backbone. So this is the most important molecule, which is actually unstable. So by adding different groups, we make it stable. But it is a basic structure, or you can say backbone of all carbamate insecticides. So based on that, what type of groups we are adding to it, what type of modifications we are doing, based on that, the classification is there. Mainly these four classes are there. So what you can remember in this classification is that some important molecules like carbaryl, carbofuran, propoxur. So this type of insecticides belongs to which class, which group, that you have to remember. So more information I have given here. There are some trade names, important trade names also you have to remember. Like propoxur, you know, it is a Baygon, and Baygon is used mostly for the cockroaches. Other insects are it is used, but for mostly it is famous for cockroaches. Then methomyl, it is trade name is Lanet. Now when I was talking about the status, you see for methomyl, this 12.5% liquid and 24% formulation are banned for manufacture. We cannot manufacture in India. We cannot import this formulation, and we cannot use. So if you cannot do, we are not doing these two things, means definitely we are not using this. But the 40% SP formulation is still in use against lepidopteran pests, mealybugs, and thrips. So this type of peculiar example they can ask you in a exam. So you should know about the Bhopal gas tragedy. It is linked to our carbamate insecticides. So what happened to that we can say the very bad night. So this reaction when it was taking place, this phenyl, when we are preparing this phenyl methyl carbamate insecticide, mainly these insecticides are more toxic or more effective, you can say. This methyl isocyanate is added to phenols. So this reaction is actually taking place when we are synthesizing the insecticide molecules. But the care has to be taken that even these compounds are reacting, water should not enter there. If water enters there, the reaction takes place and the reaction is exothermic. So it will release heat in very high amount. And when this tragedy happened, that thing was happened, somebody by mistake or by accident, the water entered into this tank, and that reaction takes place, and this methyl isocyanate gas was released, and this gas was very toxic, and you know what happened because of that gas. So in brief, you should know when this tragedy happened, what was the gas responsible for it. So this I have given, suppose when you are studying botanical insecticides like nicotine or other insecticides, I have given just in brief, you should know what is the mode of action. So they are mimicking neurotransmitter acetylcholine. Other groups of insecticides which have similar like nicotine are neonicotinoids and spinosins. That you will get in that IRAC list also. Like rotenone, what are the toxic principles from which it is extracted? What is the synergist? I was telling you about the synergist. What is synergist used for rotenone? So what is the mode of action of rotenone? It is actually a respiratory poison. So in respiratory chain, you know, this electron transport chain, it takes place in the mitochondria. So there it is affecting. Like Sabadilla, what is the active principles from which it is obtained? Then what is this mode of action? It is acting on the voltage gated sodium channels. So along with Sabadilla, other insects such like type 1 pyrethroids and DDT and related analogs, they are also acting on voltage gated sodium channels. So what happens when it is affecting voltage gated sodium channels? This much it will not be asked for JRF, but definitely for SRF and ARS, you should be clear about these things. For JRF, just till this, what is that? These insecticides are affecting voltage gated sodium channel. That will be sufficient. Then Ryanea, this alkaloid called ryanodine, which plant it is extracted, that you should know. Then what is its mode of action? You see here again, mode of action is different. So you should know this mode of action. Then synthetic pyrethroids, how these pyrethroids came into existence, what are the different generation pyrethroids, first generation, second generation, third generation, fourth generation, some typical examples, some examples like pyrethroids which are used in soil application also. So that is a typical, you will find when you are studying this, is pyrethroids used for soil application. You note down and you have to find out. So this all these details are there. You have to go through these websites. And what happened? This whatever I have given here, insecticide recommended for use, biopesticide recommended for use, and insecticide formulation banned and restricted for use. All this information I will be sending. You go through this. Then formulations I have discussed. Now in detail, when you are studying, you just go through the different terminologies I have given here. What is the emulsifier? What is emulsion? what is suspension, what is solution, what are the surfactants, that all the terms you can study. Then pesticide application equipments, definitely you might have studied in your courses. So here different equipments, what is their working principle, in which conditions they can be used, and what are the different types of nozzles, where they are used. That all the things you have to remember. Some questions I have given. Then host plant resistance, HPR, then its mechanisms are host plant resistance, then tritropic interactions, what is biotypes, you should know about the Golmich biotypes, if other insects where biotypes are important, whether we have BPH biotypes in our country or not, that also you should know, and what is the application, by how we can use biotechnology in host plant resistance. This is most information is there in triple E book, that is Elements of Economic Entomology. Class. Just 10 minutes. So just some points I have given here. They are self-explanatory. You can check. So innovative approaches for pest control. Here mostly we study attractants, repellents, antipedants. What is antipedants? They are actually not allowing insect to feed. Means we should, you know that some, if something is bitter in taste, we don't eat that. So like that, for insect also some bitter taste is there. So such antipedants are there. So for this antipedants, you should know neem as an example. Then semi-chemicals, I will give brief about the semi-chemicals in next slide. Then insect growth regulators, I discussed earlier, hormones, how we use hormones, and then insecticides based on that hormones. Then genetic control and chemostimulants, that aspect is also covered in the KP Srivastava book. And other things I have we have covered earlier. Just one thing I have missed here in your type of appliances. Here you can write down electrodrying sprayer, and another one is controlled droplet applicator. Electrodrying sprayer and controlled droplet applicator. You just find out if you are getting information is okay. Otherwise, through your teacher, you just ask me, I will provide information. So this is about this semiochemicals, insect semiochemicals. What is this? What this word means? The definition of semiochemicals. How we classify them into pheromone and allochemical, intraspecific communication and interspecific communication. Then in pheromone, what type of pheromones are there? There are typical examples. Then in allochemicals, what are the terminologies, and to which the definitions of this, and some examples you have to remember. So definitions and all I have given here. Pheromones, the some aspects of pheromones like its history, what is bombicol and all, what are two types of main types of pheromone, primer pheromone, releaser pheromone, why they are called releaser, why they are called primer, it is there in these lines. Then what are the different pheromones like sex pheromone, aggregation pheromone, alarm pheromone, trail pheromone. So sex pheromone by itself, you can say that to attract the opposite sex. Aggregation, to attract the both the sexes of both the sexes of that population. So that to aggregate at a special particular place. So you should know some peculiar examples of aggregation pheromone like our Holotrachea consanguinea. What is the name of its aggregation pheromone which we use for its management in the field? So you might have seen some management classes that we spray the neem tree or acacia tree, babul tree, with that we keep that lure, and then we spray at the night time. Why we do that? What is the role of that aggregation pheromone? That you should know. What is the alarm pheromone? Where do we find alarm pheromone? In which insects? So just I will tell you aphids. So what is that chemical nature? E beta farnesene, it is the terpene compound. So that also you know. You should know. Then trail pheromones, in which insects we found trail pheromone. All these things are given very nicely in Chapman book. So you can write down for this, you can refer the Chapman book. So some questions have been given. Then integrated pest management, in this you have studied, you might have studied economic threshold level, economic injury level, uh, how do we take decision. All these aspects are there in IPM. Now, when we are talking about IPM, different insect pests. Now, in different insect pests, when you are studying in your notebooks and from your notes and from your books, peculiar habit of any insect pest, either their arrival, their incidence season, or their feeding habit, like helicorpa, half body inside, half body outside, that type of feeding habit, peculiar type of damage symptom, like Chylopatellus, what is the symptom? Your other type of Sporoptera litura, Sporoptera frugiperda, how to identify Sporoptera litura and Sporoptera frugiperda, that also you have to study, or you have to understand. So you don't have to read or remember each and every insect's life cycle. Peculiar, definitely when you are studying in your courses, you are going through that. So peculiarity of each insect's pest, or if you take a particular crop, in that crop what are the insects of national significance, of regional significance, then their peculiar life cycle or their incidence, then comes to the management, how we manage them. So any particular practice is followed for that insect pest, which is very effective, and only it is only for that insect pest based on its life cycle. So that you have to remember. Like in rice pest, nippling or rope or alternate drying and wetting, for which insect pest it is followed. Like your blister beetles, hand collection can be done in morning. Why it is done? It is very sluggish in the morning time. So like this, some aspect or most important aspects of pest should be, you have to remember. So other things I have given here, like terms which generally you have to remember. So here I have given here the pest of different crops, and you have to see causal organism, their characters, site of oviposition, pupation. If it is more peculiar, then you have to understand, like it is lower surface of leaves, like Helicorpa, they lays single eggs on tender part. Like your Sporoptera, different species, they lay eggs in a group covered with their scales. So that is a very peculiar, because that helps for management practice also. So that is peculiar, that can be asked in an exam. Then nature of damage, symptoms, what symptoms appear in the plant. And then IPM packages, what specific practices used in IPM packages. So likewise, pest of horticulture crops and all. Then pest of store products and their management. Store products, their classification, then their management. Then about your phosphine, what is the composition of phosphine tablet, then why we use different component there. One is the active ingredient, why we use other component in that tablet, that is that you have to remember or understand. Then status of methyl bromide, whether it is completely banned or it is restriction in use, if it is in use, where it is used nowadays. And then some alternative fumigants which are not there in use, but they are potential. Some questions I have given here. Then last part, always we see this part. I think you don't have to go much deeper in any book. You can follow two books. One is Insecta for this, and triple E book, Pest of Medical, Household and Veterinary Importance. You should know all those pests they have listed nicely. You should know what type of diseases they transfer. When they transfer disease, what is the causal organism of that disease, that you have to understand, and then management. So that much is sufficient for this other the medical, household, and veterinary pest. And when you are studying the different insect pest of crops, there are different viral diseases that are transferred by that by those insects. Also, you have to like BPH. You know, Hopper-1 symptoms. Why that Hopper-1 symptoms appears? That also you should know. So like that, you have to white flies. Which white flies transmit which disease in cotton, in your bhendi, and other crops that you have to remember. Then comes to your beneficial insects like honeybees, apiculture, their life cycle, and different aspects of honeybee that are, I think, totally given in KP Srivastava book. So if you cover that much, and triple E book, Elements of Economic Entomology, if you cover these two for this, please mute. Please mute. Somebody is talking. So if you follow these two books, Triple E and KP Srivastava book, then sufficient for this honeybee. Most important part in honeybee is caste determination. What are the caste? How the caste determination takes place? Then what are the glands which are secreting hormones in case of females? That mandibular glands and all that, those details. Then pests of the honeybees, that your mite and other pests which are, and their control, that you should know. Likewise, then you have to cover sericulture also, lag culture, and other useful insect. So here I have given reference books. Mostly, if you see, these are very famous books nowadays. So this Pant and Ghai book, R.F. Chapman book, and KP Srivastava book, I think I have forgotten to mention here. So that name I will send you. That book is also important. 2 volumes are there. Current volumes you just check out. That are also important. So thank you. This much is from my side. If you have any questions, you can ask. Is the Raja is there?
I will request students doubts or questions. Then welcome and ask to any students. No.
Do our students have groups of JRF preparation? JRF.
So, Patil sir.
Okay, sir. How, sir?
Okay, sir, just 1 second. I have my MSc students also who have cracked JRF just recently. So if any students want to ask him also some questions, they can ask us. If they are hesitating to ask me, they can ask my students also.
Okay.
Students,
The participants, students, you can raise the question, please. कुणाला काही प्रश्न असतील ना विचारा, काही घाबरायचं गरज नाही. स्टुडंट्स आहे तो MSc चा. तुम्हाला जर काही हेजिटेशन वाटत असेल सरांना विचारायला तर तुम्ही त्यांना विचारू शकता काही प्रश्न.
पुढे विशेष म्हणजे आपले महाराष्ट्राचे पुन्हा घाबरत, त्याच्यामुळे घाबरायचा तो विषय
हा, घाबरायची काही गरज नाही. Anybody from out of state students, can you ask any question?
and if there are no questions, even if they face any difficulty when they are preparing, they can contact anytime in IRI. Many students are there, they can help them. There is no problem.
Yes, yes. तुम्हाला कधी काही जरी अडचण वाटली तरी सरांचा मेल आयडी ठेवा, फोन नंबर ठेवा. Whatever you face any question or queries or anything else regarding study material, you can ask for him.
Okay. So I
keep the
just a minute, sir. Thanks, sir, Nebapure sir. And I welcome Dr. B.V. Patil sir to make for the vote of thanks. Sir.
Okay, thank you very much. So I will take this opportunity to place the vote of thanks finally. So it was a very good sessions has been conducted by Dr. Nebapure sirs, and he has touched every points of the entomology. I was attended and listening him since long time. So he was given a very good and very interactive lectures and presentations. Or he has interacted on the morphology as well as the biological control, chemical control, including integrated pest management. So every point he has touched, and he has a very simple explanation has been given to the students what they needs to understand, what they needs to remember, how to go through the study for the JRF, ARS as well as SRF. So that will be definitely helpful this session for the students, those who have attended today. No matter how the numbers of the students he has attended, because several students may not be interested in the JRF of the entomology. So maybe the number is less, but whatever the population has been joined today, we can I can hope that they almost all they are interested in the entomology. So thank you very much, sir, Nebapure sir, for your very good presentation. And besides, I will also thank to our principal, Dr. Rakesh Ahire, as well as Dr. Ganesh Gaikwad sirs, who are taking the initiative to start this series and well organized session of the lectures here they are conducting for the students. So thank you very much. Besides the students, participants, they also attended the classes, and I will request you don't miss the lectures in the series. That will be very beneficial as well as helpful for you. So thank you very much.
One information for students that tomorrow we are having the lectures of 3 ARS scientists from working from right now in Maharashtra, but they will be posted in ICR. So come tomorrow at sharp 9:30. I will also send the message on our WhatsApp group. Okay, thank you. Nebapure sir, just one. Meshram sir कुठे आहेत आता? माझा काय बरेच दिवस कॉन्टॅक्ट नाही त्यांचा.
नागपूरला आहे सिट्रस मध्ये.
ओके ओके सर, थँक्यू. ओके
ओके थँक्यू. ओके.
प्रणय एंड कर मीटिंग. हेलो प्रणय.
Okay.